Welcome to Chiropractic Science, where you get to hear interviews with leading chiropractic researchers from around the world. It's been a little while since my last episode. My father passed away, and I've been busy working on a few scientific articles. I do plan on inviting more guests to the show, so stay tuned for upcoming episodes. Remember that on the podcast, you'll hear about chiropractic research from the authors in plain English, not through the media nor a middleman. My name is Dr. Dean Smith, and I am the host of Chiropractic Science. I am a clinical professor in the Department of Kinesiology and Health at Miami University, and I'm also a chiropractor in Eaton, Ohio. My research interests relate to understanding how chiropractic affects motor control and human performance. Today, I have the privilege of interviewing Dr. Carlos Gevers, but before we get to the interview, I wanted to thank all of you who have subscribed to Chiropractic Science, and I'm especially appreciative to all of you who have contributed 5-star reviews on iTunes. iTunes really helps others find out about chiropractic science. So, if you like the show, please take a second and write a review. It will support chiropractors everywhere. If you'd like to leave an audio review that I might include on a future episode, just connect on Facebook or send me an email. Please consider making a contribution to Chiropractic Science to keep these podcasts going. You can do so on our website by making a donation. All right, on to the podcast. Well, let's get on to the interview with Dr. Carlos Gevers. Carlos Gevers is a second-generation chiropractor. He graduated from Life Chiropractic College West in 2003 as valedictorian and the recipient of the Clinical Excellence Citation Award. After practicing for a few years in Spain and France, he was instrumental in the opening of the Madrid College of Chiropractic, where he still lectures. His contributions at the MCC were mostly related to the clinical sciences. In parallel, he served for 7 years as the president of the Spanish Chiropractic Association, being involved in the chiropractic politics and policy at the national and international level. In 2018, he decided to switch gears and take a completely new path in the research arena. He started his PhD in pain neurosciences at UQTR, University of Montreal Joint Program under the supervision of Dr. Matthew Pichet. His PhD studies are partially funded by a prestigious grant from the government of Quebec. His line of research and his first publications are mostly focused on mechanisms of spinal manipulation, particularly as they relate to central sensitization and neuroinflammation. These two phenomena seem to be strongly implicated in the development of chronic pain syndrome and other conditions. Well, Dr. Gevers, thank you so much for coming on to the Chiropractic Science podcast.
Thank you very much, Dean. It's truly my honor.
Yeah, so as I like to start these interviews, normally, would you mind telling us how you became interested in first becoming a chiropractor?
Yeah, of course. I think I had it easy because my dad was a chiropractor, as you mentioned before. My father was one of the first chiropractors to come into practice in Spain in the 70s. He was a Belgian national and went to study at Davenport, Iowa. Graduated in 73 and then moved on to Spain, which was still a dictatorship back then under Franco. And when he moved here, I think there were maybe 3 chiropractors at the time, but there wasn't, for example, a national association or anything. So my dad started a practice in Madrid in 1974, which became pretty big rather quickly. And then he went on to become the first president of the Spanish Chiropractic Association. So I know this is not about him; it's about me. But I think his influence is huge. It's huge on me, and I gotta say, 2 of my sisters are chiropractors as well. So we pretty much lived chiropractic since we were kids. Very early, for example, the first meeting of the Spanish Chiropractic Association took place in my living room. I must say, that was quite unique. I actually do remember that. He was a huge influence. I didn't want to become a chiropractor at first. I think, as many teenagers, I just wanted to do the opposite that my dad, showed me, or that my dad seemed to want from me. So I rather resisted But my older sister, Evelyn, she went on to start her chiropractic studies in Life West as well. And I saw her so motivated, so enthusiastic about her career that that's actually what really made me, I think, grow up and decide, okay, you don't have to do the opposite as your father to be happy, which was my motto as a teenager. And that's pretty much it. That's what got me into chiropractic, my family.
Yeah, that's awesome. I just a quick story from my end too. I was celebrating my dad's life here recently. I had a trip back to Canada, and it made me reflect a lot about his career and whatnot. And since you brought up family, he was a psychologist, and I told myself I'm never going to get into that. And and turns out that's what I got my PhD in, psychology. So I know the whole family thing. My brother's a chiropractor as well. So yeah, that's awesome. So how did you get from this first association meeting in your living room then to chiropractic school? And then tell me about that journey a little bit into practice, and all these other cool things you've been up to.
Yeah, I like to reflect on those times. Those were happy times, very, very happy. I must say that my father never really pushed us to become chiropractors, and we could see that was something that he would love, but he never tried to get us follow a specific career path. So when I went to chiropractic college, I really discovered the profession that I didn't know back then. My father had always practiced as he was a Gunstead, very classic, and we could say straight practitioner, but a very, very ethical one too. So he definitely set an example, but it was also something that was not what I would have wanted for my life. And when I went to chiropractic school, what I really enjoyed was all the neurology that was behind the mechanisms that at the time, in the early 2000s, we were starting to understand a bit more of the activation of the mechanoreceptors during the spinal manipulation, and how that activation of mechanoreceptors could get also an impact on different parts of the nervous system. So I got very quickly involved in, for example, I became the president of the neurology club that was not a thing back then. So I'm really interested in mechanisms, in neurology, in neuroscience. But as soon as I moved to Spain, you know how it goes. Yeah, clinical practice can really get overwhelming in a good and in a bad sense. You get good response from patients. You can make a good living out of it, and you feel like you're doing something useful for society. So I was really focused on practicing for a few years. I opened up a practice with my sister in Madrid. We were working in one of my dad's practice he had in the north of Spain, in Bilbao. And then I also moved to France to practice a bit, but that's another story. It's related to a woman that I met back at the time, which is often the case. So I also practiced in the French Alps for a couple of years. That was also very, very interested. And it wasn't until then that there was no chiropractic education in Spain. We're talking 2006. But at that time, the first talks that seemed to be getting somewhere with a university is the place where I'm currently still lecturing in Madrid. They seem to be reaching a point where there was going to be a chiropractic program starting in Spain, which it ended up happening in 2007. At that time, I was in France, but I was very, very interested in being able to contribute. So I decided to move back to Spain, and I contribute to the project from the early beginning, slowly finding my place where I could also contribute teaching. I was also doing some of the administration necessary for the opening of the school. And that also got me into politics, because obviously the Spanish Chiropractic Association was instrumental in the opening of the college. You need resources, people who can teach, not just chiropractors, obviously, mostly non-chiropractors, I would say. and you need a lot of economical resources. So I also got involved and was elected secretary of the Spanish Association also in 2007 when I started. I started also teaching in the first cohort of chiropractors in Spain. Yeah.
That's a lot of stuff.
It is a lot of stuff. I was very young too. When I reflect back, I was 26, 27, and like, wow, I was really a kid. And I thought I wasn't, but now looking back.
So you've got this interest in neurology. You're teaching. You're involved in politics. So what propels you from that point to get involved in research? It seems like your plate is kind of full.
Yeah, you're right. And I honestly don't know how fun time, but the thing is that I think for some of the listeners that may not know, chiropractic is not regulated in Spain. It's obviously not illegal. There's just nothing that regulates chiropractic to this day. There are 2 chiropractic education programs in Madrid and Barcelona. None of them are recognized by the Spanish government, although they both have CCE accreditation. So it's quite a special situation, which means that everything is very entangled here. Like, okay, you want to teach? Yes, well, you must get involved with politics because the regulation of the profession depends first on the regulation of the chiropractic education. So this is also, I think, one of the reasons why I had so much in my plate. I think I had the capacity and definitely the energy to help. But as I was progressing, as the program was progressing, we got our first accreditation in 2012. Actually, in 2012 we got the first accreditation. Then we got reaccredited in 2015. So it seems like the program in Madrid was getting established, but I felt like I was getting stuck. I was teaching, but I was not developing my own knowledge. I was not growing as a lecturer, as an instructor, and that's when I realized that I had to follow the classical. Maybe I don't know if the tenure track path, but I had to follow the classical path that you need to follow in order to become a professor or at least an academic. We had to do it in an informal way because one of the barriers that we have in Spain, I cannot undertake a PhD program here because my diploma is worth nothing, pretty much. I was able to get into a master's. So I took a master's of neuroscience program in 2012. They accepted me into that program. I had my dissertation. I passed it, but then I tried very hard to get into a PhD program, but they rejected me based on the fact that my undergrad training is not recognized. So it's as if I hadn't taken any undergrad training. So eventually I realized, okay, I need to find a way outside of Spain that I can continue training, that I can become an academic, and I can not just teach, but also create knowledge, contribute to knowledge production, and become a scientist. Yeah.
Yeah, that's really fascinating. I had no idea about the, your issue trying to get into a PhD program there in Spain. Wow, that's really fascinating. And then also just the struggle of I don't know if struggle necessarily, but the challenge, let's say, of trying to get a PhD outside of your own country. Yeah. So tell us a little bit about that. How did you get to UQTR, and what's it like? I'm also interested if you're still in practice too, as you're trying to do all this stuff.
Well, just telling these stories, I feel exhausted already. Like, how the heck am I doing this? But, so I just turned 40 last year, and I think, I was able to do it because I was younger, but I'm getting to a point where I am actually at the point where I'm making choices. So let me rewind a little bit. So I was looking for options to undertake a PhD program. We had a collaboration with a professor from RMIT in Australia, Dr. Michael Lazari. He's a chiropractor over there, Australian chiropractor. So we collaborated with him and the University of Sevilla in the south of Spain for a project with chiropractic and podiatry treatment for low back pain. And he was actually the one to mention me, okay, maybe you should actually get a PhD, and this is the way for you to go and become an academic. I explored the possibility of Australia, but funding was not easy at the time. So what happened is that UQTR and Madrid College signed an agreement right around that time. I think it was 2017, an agreement for exchange of both students and faculty. And talking to who is now the person who is now my co-supervisor, we found that this could be a way for me to undertake a PhD program in Quebec. So I was interviewed by my current supervisor, Professor Mathieu Pichet, and he was kind enough to accept me in his lab after I got an interview with him. And we started moving everything so that I could go to Canada when I wasn't teaching here. That means around 3 months during the summer, and maybe 2 more months, for example, during spring break, Christmas break. And I've been doing that since 2018. Unfortunately, something happened in 2020. I don't know if you heard of it, the coronavirus pandemic.
Never heard of it.
Yeah, I wish, right? So that made it a bit more difficult, and we had to rearrange everything. And I've been doing actually all my data collection in Spain since, and I haven't been back to Canada for longer periods since the pandemic. But now that almost all my projects are finished, I am ready to move over there, write my thesis, defend hopefully next summer. And I'm currently in search for a postdoc in Montreal. So this is where I am right now. Yeah, and actually, let me also answer another of your questions. I am still seeing patients. I am still seeing patients because, as you may know, academia doesn't pay very, very well. So I am still in practice. I mostly do it I mostly see patients that I have seen for years, just as part of their maintenance care or their chronic conditions. I do enjoy it, and I think it's necessary. But if I'm very honest, it's also an important part of the income that I'm getting at the end of the month.
Yeah, no, definitely. And I think a lot of researchers end up having, multiple interests, teaching, doing, seminars, continuing education, this sort of thing. There's lots of avenues, and maybe we can explore that a little bit later when we give some advice to, people that may be interested in research as well. But let's get into some of your studies now. You've done a lot of cool work on the mechanisms of spinal manipulation, various other topics. I'm excited to talk about all these different studies. And the first one that we can talk about, I think, might lead us down the pathway of talking about the other studies as well. The first one is neurophysiological mechanisms of chiropractic spinal manipulation for spine pain. So, most people, I think, all over the world probably know chiropractors for spine, and they consider us when they have pain. I'd love them to come in for performance and all sorts of other things, but pain is definitely the main driver for why people come in. And so if you could lead us through this paper, and I think you've done it in a very fascinating way, one that I particularly like, and that is that in the paper, you're classifying pain relief by different mechanisms. And so I'll just briefly cover these, and maybe you can talk about them in a little bit more detail. But different mechanisms, including pain relief from peripheral mechanisms, the spinal cord, supraspinal, and also placebo mechanisms. Also a nice touch there. So I think it would be beneficial for chiropractors and patients, whoever else is listening, if you could summarize some of these key points within each of the ways that you've classified this pain relief.
Thanks very much for those comments, Dean. That's very kind of you. This is it's a paper that's very dear to me because especially during the time that we were unable to do much testing, I dedicated a lot of time, and it is actually it's a great work of learning when you're doing a review like this. This is my first attempt to a narrative review. It's not a systematic review to start with, but we thought that this was a more appropriate design, and so did the reviewers, so that we could tell what has been published, not in a systematic way, but choosing specific topics. And this is one of the keys that you mentioned, right? How can we try to understand what is happening when somebody is getting a chiropractic adjustment or a manipulation that leads them to feel less pain? So when we're looking at the evidence, there's, these recurrent topics kept coming. So there are things that we can measure in the periphery that we classified as peripheral mechanisms. By the periphery, usually we mean, for example, serum samples. So something that you can measure in the blood. You don't really know where the mechanism is taking place, where the effect is happening. Sorry. But you can definitely say that there is something happening to peripheral biomarkers. When we're looking at peripheral biomarkers, we found that there's a lot of literature, but not always very consistent. For example, there's a lot of literature on cortisol changes after a spinal manipulation, but some of them seem to suggest that cortisol increases. Some suggest that cortisol decreases. We don't know if maybe those changes are related to a non-specific effect due to the fact that, for example, when we get a neck manipulation, you can have an acute perception of danger. So you have a stress response that could increase your cortisol levels. And this could contribute to pain relief, but that wasn't always consistent to the responses found in all the studies. So I don't think we could say that we know what happens with hormones, and in particular cortisol, after spinal manipulation, and whether they contribute or not to pain relief. We did find a few studies that looked at two things that we related. Cytokines and chemokines, that are substances that are released by the immune system, and they seem to regulate, in the studies that were linked to spinal manipulation, they seem to regulate the inflammatory response. And also some studies, both animal and human studies, that found changes in reactive oxygen species. So what does this mean? It was a bit hard for me to understand, but it seems like through the spinal manipulation, there is a decrease in peripheral oxidation. And how could this happen? We related that to inflammation. We know that inflammatory processes lead to increase in peripheral oxidation. So we believe that these two topics are probably related. And we don't know the exact mechanisms that begin this, but we can measure in the periphery changes that tend to be anti-inflammatory after spinal manipulation. So I hope I made this somewhat clear. It seems like we could measure something in the periphery, suggesting that the manipulation leads to less inflammation and also less pro-inflammatory responses. So overall, reducing the inflammatory response. But these were all usually small studies, some of them in animals, so we can't really extrapolate that to humans. Some of them were both in animals and in humans. I think it's still early to draw conclusions there. But what we found more interesting, as you were mentioning, we divided in mechanisms in the periphery, and the biggest section of the paper was related to mechanisms in the spinal cord. And it seems that most changes that are related to pain relief after spinal manipulation act through the spinal cord. This is where most of the evidence is pointing. Do you want me to keep going, or do you have a specific?
Yeah, no. That was one of my questions because I think when I teach continuing ed and or I'm a participant in continuing ed, I get the sense of we get all these, we'll say, instructors that say, well, we adjust and then it goes up to the brain and they draw all these pathways and talk about chemicals and things. And I think to myself, okay, yeah, some of those seem like those are the neuroanatomical pathways for sure. But where's the evidence?
One of the reasons is it's very hard to measure. It's really, especially in the human, it's very, very hard to measure.
Exactly. So that's all I was going to say, and I'm glad you mentioned that. Yeah, the strongest evidence, or the most evidence, we'll say, happens to be for the spinal cord. So yeah, please tell us about the cord. So
I think, I don't know if I could say it's the strongest evidence, but definitely the largest volume of evidence, because there are spinal mechanisms that can be, let's say, easily measured in humans. One of the mechanisms that has been measured repeatedly by the team of Bialowski in Florida, that you may be familiar with. I'm sure you are, actually, Bialowski and Bishop, but also from the lab of my supervisor in a paper on 2017, is the temporal summation. So the temporal summation of pain is a phenomenon that is known to happen in the dorsal horn of the spinal cord. We know that when you induce a train of stimulation and you compare it with a single stimulation, this tends to create either an enhanced pain response, or an enhanced pain perception. And from animal studies, we think that this happens because C fibers have this intrinsic property where they summate on the cord, and it amplifies the message. So you can test for a temporal summation by using, for example, repeated trains of electrical stimulation, and compare what this provokes to a single stimulation, right? So you can see how a person reacts, or what intensity of pain he or she feels if you have one single stimulation and compare that to multiple stimulations at the same level. For example, with electrical stimulation, but you can also do it with thermal stimulation using a thermode. And there's a few studies that suggest that spinal manipulation, rather than decreasing a single thermal stimulus or a single electrical stimulus, it acts by decreasing the temporal summation of those stimuli. And we know that this is something that most likely happens, as I was telling you, in the dorsal horn of the cord. So, there's about 5-6 good papers that we can refer to in that sense, and I think these are important for our listeners to be aware of. And apart from that, there's a few papers that have been looking mostly at pressure pain thresholds that you can easily measure with an algometer. And they've measured changes in pressure pain thresholds before and after spinal manipulation in the same segment of spinal manipulation was applied. So when we're talking about the same segment, is either right by the spinous process, but also in the same dermatome. And there's actually quite a large group of Spanish investigators. One of the main ones is Fernández de las Peñas. And they've reproduced these findings in the myotome, in the dermatome, mostly addressed at the cervical spine. So when we see a change that seems to be specific to the segment, you can also assume that the changes are happening in the spinal cord, in the same segment of the spinal cord. However, this is what we found, and this is what we concluded in our review, that there seem to be segmental changes after spinal manipulation in the spinal cord, mostly related to the temporal summation, so likely related to the inhibition of those C nociceptive fibers. But this is currently a very important debate. I don't know if you've read, there was a very recent review by Casper Nim, and Scientific Reports 2021.
Oh, yes, about the location.
Exactly.
Yes. Yeah.
Exactly.
Yeah.
So in this paper, they were looking at whether the effect of a spinal manipulation was different when you apply it at what they call the candidate site versus the non-candidate site. So like, are we really specific? And they concluded from those papers that the segment of application didn't matter. This is in contrast to what was found by another review that we referred to in our paper, and by the conclusions in our paper. That review, if I'm not mistaken, was published by Margot Honoré in 2018, I think, in Chiropractic and Manual Therapies. They concluded that, when we manipulate a patient, the changes in pressure pain thresholds specifically were happening at the same segment of application. Those were the strongest effect sizes that they found.
Carlos, I'm really glad you brought that up. I was just teaching a continuing ed course last night, and I reviewed a couple of animal papers. And in the literature on cats, it seems that, they adjusted L6, I think, in these two papers, which were the target vertebra. And then they adjusted L5 or L4 as well as L7. And they were specifically looking at the type 1A sensory neuron. So coming from a muscle spindle. And what they found was that the target did matter, that they, at least from a neurophysiologic perspective, that it stimulated the type 1A sensory neurons a heck of a lot more than at a non-target location. And I wonder if some of what we're seeing here, is difficult, as you said, to get the effect in humans, or to be able to at least study or quantify this type of effect, versus when you do a little bit more of a classical, let's say, mechanistic animal study, we're seeing larger effects.
Yeah, this is the work by Dr. Reed, right? Dr.
Exactly.
Reed.
Yes.
Yeah.
Yep.
Fascinating. I love that. The thing is, I love mechanisms, but, is this first of all, is the stimulation of muscle spindles relevant clinically? We still don't know. Obviously, we have, like you were saying before, the neuroanatomical pathways that, could give some plausibility, but we don't know if it has any clinical relevance. Just like we also don't know if pressure pain threshold changes, for example, or changes in temporal summation. We don't know yet if those changes that seem to be induced by spinal manipulation have any clinical relevance. So it's not just difficult to translocate to humans, but it's also very difficult to know if there is if it has any impact on clinical outcomes. Actually, there's another cool paper by Nim, Casper Nim, also on Scientific Reports. This was 2020. And I found this very interesting in contrast to their systematic review, because they found that doing the adjustment, the manipulation on the most sensitive segment, as measured with an algometer, didn't change the clinical outcomes. So that's exactly what we're talking about. But it did have a specific impact on the changes in pressure pain thresholds, as opposed to doing the adjustment elsewhere. So what they concluded is that the neurophysiological effects seem to be segment specific, but the clinical effects did not seem to be segment specific, which is puzzling, to say the least.
Yeah, for sure.
That's something we couldn't answer in our review. It seems that, the adjustment, the manipulation works through segmental mechanisms, but, how relevant are those mechanisms for patient pain relief? Because also we were reviewing specifically for this study. Some of the studies use clinical populations, but they were interested in mechanisms. So we can consider that this is a review of basic science studies,
Yeah. And I will definitely need to get to the supraspinal and placebo and wrap this paper up. But I just want to throw in some of the studies that my colleagues and I have been looking at, we're actually not interested necessarily. Well, we are interested in the mechanisms, but we're looking simply at the behavioral outcomes. What do we observe if we adjust this or that? Is there a difference between adjusting one segment versus another segment on behavior? Let's say balance or postural stability, something like that. And so we're noticing changes depending on where and what. Let's say upper extremities versus lower extremities. Some of the studies that we've done, and so you're right. There are so many different angles to get at this. Somewhere in there, the clinical outcome obviously is important to the patient. Pain relief, as we're talking about in this particular paper, but other behaviors also I think are important there. So it's wow. There's so many ways, that, like I said, to come and approach all of these issues. The neurophysiology has got to be important at some level, but the translation into clinical practice and what the patient gets out of it has got to be where it's at. And I don't know. What are your thoughts on that?
I'm going to tell you something I read this morning on Twitter. It's a physio that I follow that I really like. I really enjoy what he posts, but very critical of manual therapy. And he was talking this morning about what he called mechanisms masturbation. So how many researchers we like to just enjoy and try to unveil mechanisms that may not be clinically relevant. And I agree in a part, but I strongly disagree in another part, because until we don't understand the mechanisms, it's going to be very hard to understand which effects are specific and which effects are not. Also, if we don't understand the mechanisms, it's hard to even design a placebo, because when we know how something works, you can make sure that whatever that thing is doing to work is not part of the sham intervention. But so far, we don't know. When we design a placebo spinal manipulation, which is also one of the things that we discuss in this review, we really don't know what are the active ingredients of spinal manipulation that should not be part of it. For example, there are papers that seem to suggest that there's, the cavitation has no impact whatsoever. So, that doesn't seem to be an active ingredient. But for patients, it seems to be very important. So if cavitation is present or not, is that going to change the placebo perception? For example, I'm just talking about something that is, super common in clinical practice. If it doesn't pop, the patient's like, oh, but you didn't get it to move. So I don't agree with him. I think there's a lot of, in science in general, mechanism, onanism, we could say. But I think this is so necessary for interventions that we're just starting to understand how they may work.
Yeah. Yeah. Well, and once you see in our research, once you see the behavior, I mean, we're always trying to link it to mechanisms for sure. And so it's just not the type of research that I've gotten into. That's why I'm so fascinated by it, because I'm trying to understand, because then if I understand the mechanisms, then to me, the behavior should be sort of a natural expression of that.
It should follow. It should. Yeah. Exactly.
Yeah. So this is an awesome discussion. So let's, if you can tell us about the other two components of this paper, the supraspinal and then placebo.
I think they're very related, because everything, let's say above the cord, is supraspinal, but also many of the placebo mechanisms, at least that we know of, seem to act from supraspinal levels and down, from top-down mechanisms. I think I really am fascinated by the research looking at supraspinal changes after spinal manipulation. I don't know if you've seen some of the papers were reviewed by Ellingsen, for example, or Weber. Carl, I think is his first name. Carl Weber.
Ken Weber?
Ken
chiropractor?
Weber, sorry.
Yeah.
Weber,
From Stanford.
Yes.
Yeah.
Great, fascinating stuff. The thing is that, none of these studies can conclude that the changes they see in the brain are the direct consequence of a spinal manipulation on the brain. but rather, it could be an indirect effect of spinal manipulation changing nociceptive transmission, even as far down as the cord or even in the periphery. So if you change the ascending pathways, you will change the reactions and the brain areas that are activated. So we can't really conclude that there's any mechanism that's happening, let's say above C1. We can't really, at least for pain relief, it doesn't seem like. There's also a review that concluded that we mentioned in a study. I'm sorry for the first author. I forgot. I think her name. I know Charlotte Leboeuf is in that review. They were reviewing functional neurology and
Oh yes, I remember that paper. Yep.
Yeah. So I don't think we can conclude that as much as I wanted to find something like that, because I did do the CARI postgraduate training. I did it twice. I found that fascinating, very interesting, but there's just not the evidence to say that yet. We can say that there are definitely contextual effects and expectations that are influencing our outcomes. Some studies, there's an interesting design by Bishop and Bialowski again, who look specifically at expectations, and they separated changes, or they try to separate it, what changes could be attributed to expectations. It's a big component of the placebo effect, expectations. And they looked at changes that could be independent of expectations, and they seem to find, they concluded that after spinal manipulation, there were changes in the temporal summation that could not be attributed to the expectations of pain relief that were reported before the experiment. So, there are definitely contextual effects, and I think they're very strong in any manual therapy. But that doesn't mean that the contextual effects can explain all the changes that we see in our patients. One of the main weaknesses we already mentioned is that designing, and everybody that has read a single paper on spinal manipulation, they know that designing a placebo spinal manipulation is very difficult. It's very difficult because you can follow on the two extremes. You can have an intervention that doesn't look at all like spinal manipulation. For example, some studies have used rubbing on the spine of the participant, like a very light effleurage massage kind of. And other papers have used very, very similar procedures that they look so similar to spinal manipulation that they actually may carry some of the effects. So, on one side, your patient is not blinded because they know what you're doing is nothing like a chiropractic adjustment. And on the other side, the patient is blinded, but your intervention may actually carry some effects. So it's very difficult to sort out what are contextual and what are non-contextual effects. But it's not just for spinal manipulation. I want to say that pretty much every physical intervention, massage, exercise, mobilization. Exercise is a big one. The placebo, the sham exercises that are used in the different trials are not very strong. It's difficult. It's just difficult to do.
Yeah, definitely.
Is this answering?
Yeah. No, this is excellent. So then, we've gone through some of the, we'll say, more hardcore science to this topic, but I'm also interested in, obviously, the translation to the chiropractor and to the patient. So one of the ways maybe that I think would help chiropractors is to hear you've done the research, and you're a teacher as well. How would you translate this body of evidence, these four mechanisms, in talking with patients in your practice? I'm just kind of curious of how that conversation would go. Are there some key talking points that you might use with patients to describe how they may achieve pain relief through an adjustment? If you could help us with that, that would be awesome.
Mm-hmm, mm-hmm. Yeah, that is, I think, this is the $1,000,000 question, right?
Yeah.
How can you really extrapolate that? First of all, I think what's important is to acknowledge that there is a trend. The trend has been there for more than 20 years now in the literature, suggesting that our adjustments and manipulations are not as specific as we thought they were. The biomechanical changes that we thought or we still think we make, at least there's not much evidence or not very compelling to support that there are relevant biomechanical changes, at least specific biomechanical changes after spinal manipulation. But I do think that there's more and more neurophysiological mechanisms that are unveiled. Some of them may prove to be relevant for the patients in the medium long term. Some of them will not. But I think it's important that we explain our patients that, I think this is current best practice, that we're not really changing very much the mechanics. We're just putting an input to the system. It's a neurophysiological input. Most likely, we could also talk about the studies by Reed et al. That you were mentioning before, and actually there are some papers by the Nougaro, and a lot of papers were led by Martin Descarreaux in UQTR also, where they couldn't measure the 1A fiber activity, but they could measure the muscle reflex that's provoked most likely by the activation of the 1A large diameter fibers. So it seems like we do activate peripheral fibers, and we create a non-nociceptive input to the system. And I don't know if the patients will be very interested in this now that I'm listening to my own words. But I think it is important to change the way we explain it. We're not really moving. We're definitely, I think this is quite old. We're not putting bones back in place. We're putting an input to the system that may have an impact on neurophysiological mechanisms that are related to pain. And the second thing I would maybe tell you is that this is something that has not been done, as far as I know, in chiropractic education very consistently. But I think we need to introduce at least a minimum of patient pain education in our interventions. Although, I don't know if you've seen, there was, if I'm not mistaken, a recent systematic review that found no changes after introducing patient education, no changing in outcomes in patients with low back pain. Still, I think it's important for our patients to understand a bit better the pain mechanisms, why they perceive pain, so that they move away from that model where pain means there is a tissue damage clearly in their low back. That tissue damage has to be detected by some type of imaging, and that it has a mechanical solution, which is either putting something back in place or surgery. I think the research that we have summarized is going in this direction. No, this is not happening. However, we can affect neurophysiological mechanisms of pain transmission and perception, and those changes, will happen at different levels that we still don't understand. I think it's too early to tell the patients, this is what's happening with the adjustment for you. I want to say, especially if we now move on and talk about the other capsaicin paper, I think there is evidence suggesting that there is some degree of specificity of the spinal manipulation. We just don't know if that specificity changes the clinical outcomes yet. We don't know that yet. So this may be another lesson that we can draw, and that maybe we could tell patients, where I adjust you is important. We don't know yet how important it is and to what means, but may be important.
Yeah, and you brought up a lot of great other issues too. The sound that the cavitation or the pop that that people hear and feel when they get adjusted. When you're talking about placebo, it made me think of, when we interview people, motivational interviewing, and, we can then incorporate just the whole experience that people get when they see a chiropractor. Probably they're going to get exercise. We talked a little bit about exercise too, how important that can be, and eating well, lifestyle, all the things that they can do. We may not know everything yet as to how the mechanisms work, but clearly something is working, and how couple that with all the behavioral aspects and lifestyle and whatnot.
If I may, Dean, I may,
yeah.
I think you brought a very good topic, a very good point by saying that as chiropractors, if we're applying spinal manipulation without thinking or introducing thoughts to our patients that we're putting, bones back in place, or, some of that, old beliefs. We recommend them exercise and that they stay active. And we're providing them with up-to-date patient education. We really are, front line for managing any type of, not any, but most types of spine care. We are obviously trained to also detect the ones that we cannot manage and we need to refer out. But, with these interventions, we're really following most, if not all, clinical practice guidelines.
Yeah, I think that's a beautiful way to put it. That's sort of what I was thinking, but I'm glad you stated it much more clearly than I did. So thanks for that. Okay, let's get into the next paper, which is Chiropractic Spinal Manipulation Prevents Secondary Hyperalgesia Induced by Topical Capsaicin in Healthy Individuals. So yeah, if you could tell us about this paper and then how this is a continuation essentially of what we've just talked about, and what it may add different.
Yeah, thanks. I'm glad to talk about this paper because also yesterday, or maybe the day before, I read on Twitter again. There's been on the JOSPT journal, the Journal of Orthopedic Sport Physical Therapy. They have a blog, and there was a specific blog post that was talking about, was it time to conclude or to assume that manual therapy was not specific at all. And they were citing this paper and a couple more papers from our lab. In my opinion, if they eventually let me have a rebuttal, in my opinion, they were not interpreting correctly our findings. So I think it's a good opportunity. Hopefully somebody would listen to this and maybe let me know if I can write a rebuttal to that. So we wanted to look again at spinal mechanisms specifically, and there's a line of research that my supervisor Pichet has been following since I would say since 2017, based on the hypothesis that spinal manipulation has an effect or can modulate the activity of C fibers, nociceptive C fibers. So in this paper, it is true there's a big limitation where it's an experimental pain model. So we are inducing experimental pain to healthy participants. And that is always a limitation, but it is also very useful to understand mechanisms, and it's very commonly used in pain research. The pain model that we use here is the application of topical capsaicin. And, I think most of us have heard of capsaicin, but just in case, it's the active ingredient of chili peppers that give chili peppers the hotness, the spiciness of it. And it does so by activating a type of receptors that are called TRPV1 receptors. They're also known for being very important for thermal perception. I don't know if you know this, but last year the Nobel Prize, Medicine Nobel Prize, was given to actually the work done on the TRPV1 receptors, the capsaicin receptors. So it is a hot topic.
No pun.
Better said. Yeah. So what we did, we're creating pain to these healthy participants around the T5 spinous process. We chose T5 just because it's an easy area to apply manipulation, not for any other, let's say, segmental reason. So we applied capsaicin around the T5 spinous process. We were measuring brain activity. We were asking the participants to rate their perception of pain and unpleasantness related to capsaicin, because we know that the activation of C fibers is also related to the emotional aspects of pain, like unpleasantness, not just intensity. And also, before and after the application of capsaicin, we measure pressure pain thresholds, but not inside of the area of capsaicin application, but outside. We're measuring, if I remember correctly, 2 centimeters outside of the area. And this is important because when you apply capsaicin, what we know that there's an activation of C fibers mostly, not just C fibers, but predominantly C fibers, which are related to these TRPV1 receptors. So capsaicin activates these TRPV1 receptors, which activate C fibers, and these C fibers stimulate then the dorsal horn of the spinal cord, and there's some changes that happen with the summation of these C fibers. Through animal models, we know that this activation of C fibers induces what we call long-term potentiation. You may be familiar with LTP related with memory.
Yes.
So long-term potentiation is key in memory processes in the hippocampus. But we also know that LTP is important, long-term potentiation is important for processes related to chronic pain in the spinal cord. And C fibers have been strongly correlated with these changes in the spinal cord. So, there's a bunch of research suggesting that capsaicin can induce long-term potentiation in the cord, and that these changes result in some behavioral changes in the pain perception of the participants, or potentially in patients as well. What happens with these changes in the cord is a phenomenon that is called primary and secondary hyperalgesia. We were interested specifically in secondary hyperalgesia. So let me see if I can make this clear. So when you apply capsaicin in an area, that area becomes painful. This area where you have applied the capsaicin is the area of what's called the primary hyperalgesia. So that area becomes red, there is a vasodilation, and we think that there is an increased perception of pain in that area due to peripheral sensitization. So the receptors become sensitized, and patients or participants start perceiving pain where capsaicin is applied. This is similar to what happens to us when we eat anything that has capsaicin, like chili peppers or jalapenos or whatever. But what we also know is that when you apply capsaicin in the skin, topical capsaicin, there is an area that develops outside of that primary hyperalgesia that becomes sensitized as well. This area is the secondary hyperalgesia, the area of secondary hyperalgesia. And this area becomes sensitized specifically to mechanical stimulation. So there is a mechanical hyperalgesia that develops outside. Do you think that's... I made this clear for now?
Yeah, absolutely. I get it exactly. So around where you put the capsaicin, that's the primary area, but just beyond that, there's an effect, and that effect that let's say lowers your threshold to pain is the secondary area.
Exactly. So often in human models, you don't perceive pain in that secondary area, but just as you mentioned, your thresholds are decreased and your sensitivity is increased. We don't know exactly in humans because you cannot measure, but from animal studies, this seems to happen because of changes in the spinal cord. These changes that I was mentioning before, summation, maybe long-term potentiation, maybe disinhibition also of inhibitory interneurons, but in humans, we simply don't know. So this area of secondary hyperalgesia, we measured it by assessing the pressure pain thresholds at the beginning of the experiment, before we applied capsaicin, and after the 40 minutes of capsaicin application. Okay, so we could have an idea of whether those thresholds that were 2 centimeters outside of the capsaicin area were going to change or not. And this was actually our main variable of interest because we hypothesized that spinal manipulation, because it could inhibit the activation of C fibers, it could inhibit the development of secondary hyperalgesia. So what we found, I think it's very interesting to say also that we found that the participants were rating their pain. And I forgot to mention that we split our participants in 4 different groups. They were assigned randomly. One group received no intervention at all for 40 minutes. So we just wanted to, with this group, we wanted to assess for the non-specific effects of time, simply the passage of time. Another group received spinal manipulation at T5, so the same level where capsaicin was applied and where these changes were supposed to happen. Another group received spinal manipulation 4 segments away, T9 instead of T5. We wanted to see if any potential changes were dependent of the segment of application. And then finally, another group received what we labeled placebo manipulation. It wasn't a great placebo because we used the dynamometer to apply a force that would be equivalent to spinal manipulation. But, it was at the time what we found the best to see if the activation of mechanoreceptors, similar to what would happen with the spinal manipulation, would be responsible for that effect or not. So, the first thing that we found is that it didn't matter if they received an intervention or no intervention, T5, T9, the way they rated their pain intensity related to capsaicin was not different between groups. So all participants reported pain after around 10 minutes on average, peaked around 32 minutes, and then decreased a little bit. So their primary pain, which is the pain that they perceived from the area of capsaicin, was not affected by spinal manipulation. And, we were not really sure that this would happen, but, it was partially expected. We were also measuring brain activity. We're measuring gamma band oscillations. Gamma band oscillations have been, lately in the last 10 years, have been linked to ongoing pain, both in experimental pain models, for example, with capsaicin, capsaicin and heat, also with heat pain, but also in patients with fibromyalgia and chronic back pain. There is an increase in gamma band activity, mostly in the prefrontal cortex. So we were interested in this to see, okay, is this supraspinal activity change after spinal manipulation? We found no change. So no change between groups, no supraspinal changes. But what we found that, this was our main hypothesis, and this is what gives sense to this paper, was specifically related to the pressure pain thresholds measured before and after the capsaicin, and also before and after, obviously, the interventions, because the interventions were provided after 20 minutes. So the experiment lasted 40 minutes with capsaicin the whole time. And in the middle of the experiment, after 20 minutes, we applied the interventions for the groups receiving intervention. So what we found is that the participants receiving no intervention or receiving the placebo manipulation with the dynamometer that I was telling you about, both of them developed secondary hyperalgesia, which means that their pressure pain thresholds were decreased at the end of the experiment. So this is expected, right? So you apply capsaicin, and outside of the area of capsaicin, you have an area where we know your mechanical thresholds will drop. And this happened to these 2 groups. But what is very interesting is that the groups receiving manipulation, both at T5 and T9, they did not develop secondary hyperalgesia. Their thresholds didn't change significantly. Actually, for the group receiving the manipulation at T5, the thresholds even increased a tiny bit. It was not significant, but it definitely did not drop. And this is the main finding. So we can say that the groups receiving manipulation did not develop secondary hyperalgesia. How did that happen? We don't know, but we hypothesize that through an inhibition of the activity of C fibers. The capsaicin was activating the C fibers, but by applying our manipulation, we decrease that activity and that summation in the spinal cord, probably.
Okay, so now I have some now I have some questions.
That was heavy.
Yeah, no, that was heavy, but it was good on the mechanism. So getting back to the review paper, you talked about temporal summation as one of these sort of key variables, we'll say, at the spinal cord level. And I'm going to do a little extrapolation here. You tell me if I go off the deep end. But so if I understand this correctly, the adjustment or the manipulation seems to modulate this temporal summation. And if that's true, and from what I understand, this secondary hyperalgesia and subsequently central sensitization may be something that an adjustment or a manipulation could impact. And then if that's true, then I'm also wondering about the gamma oscillations that you measured through EEG, and I'm wondering then if someone comes in for care for weeks on end, let's say, a few weeks of adjustments or manipulations, would that gamma band change over time as you continue to modulate that temporal summation?
There's two things I want to point out. First of all, especially if my supervisor listens to this, because I need to clarify that with capsaicin, we can't say that we are measuring temporal summation.
Okay.
It's a distinct mechanism, but it has in common that there will be an ongoing or repeated activation of the C fibers. With the capsaicin, we do know that it is a good surrogate experimental model that imitates central sensitization. So you were right about that part. So we know that capsaicin is used as a surrogate for central sensitization. And it does so by, repeatedly or in an ongoing fashion activating those C fibers. So it's very similar to temporal summation. We know that with central sensitization, the temporal summation is enhanced and prolonged. So it's like these responses are enhanced and prolonged. And this is what happens with capsaicin. So, it is somewhat related, but we weren't measuring specifically temporal summation. We weren't. We could say that these results suggest that, a spinal manipulation actually maybe could prevent the development of central sensitization. Once central sensitization is in place, we would need to have maybe a longer time period to assess this, or measure surrogates. Central sensitization is kind of a controversial topic. Not everybody in the pain community accepts that. Outside of the lab with animals, we can talk about central sensitization. So we're just going to assume that we're talking about the processes that are surrogates in the human. But, in the clinical population, we can measure some things that seem to be relevant or seem to be related to central sensitization. And it would be interesting to know if those things change specifically with spinal manipulation. We don't know yet. This, as a basic science study, suggests that this may be one of the mechanisms, but it may suggest more that it could prevent that development. So, like in the transition towards developing central sensitization, which is a mechanism related to chronic pain. More than once it's installed, can we revert it? That's a bit of a more difficult question. Also related to the gamma band oscillations, honestly, yes, our experiment was very short. One of the limitations that we had is that capsaicin creates the reaction to capsaicin is very diverse. So for behavioral studies, it wasn't so bad, but we had a lot of inter-individual variability for the gamma band oscillation. So we don't know if there could be an effect there. It would be very interesting to see this in a clinical population, which is even more diverse, honestly. But it would definitely be interesting. It's just difficult. There's an important limitation, which is the fact that when you're measuring brain activity, the participant shouldn't move for as long as possible. So 40 minutes was the length of our experiment, which wasn't long enough, I think, but it was definitely too long for participants to stay without moving. And you need to be able to do the manipulation without them moving for too long also, or too much, which is also why we chose prone manipulation, instead of a side posture that, would be very interesting for low back pain patients. It's tough because then you're moving all the cables, you're moving the cap. It's going to be more difficult to measure EEG activity in that case.
Yeah, totally. Makes sense. And I realize as scientists, we don't like to, go beyond the data. I'm totally into that, but I also do like to spend some time and hypothesize about what the implications could be, and then it gives further ideas for other studies that certainly can be done as well. And so it makes me think, okay, how many people with chronic back pain is central sensitization a mechanism? Is it everyone? Is it some people? How common is this central sensitization? And if these results that you've achieved are indicating, okay, it may not be the exact mechanism, but is it a component of a mechanism that may prevent central sensitization? I mean, that's exciting to me.
Yeah, to me too. Maybe it's mechanistic onanism, as we were saying before, but I would take that.
Yeah, absolutely.
It's a very good question that you're asking me, because this is also a very, very hot topic currently, which is we're trying to classify chronic low back pain patients according to mechanisms involved. There's been huge efforts directed in that sense since I think it's been like 12 years. There's a seminal paper by Smart et al. I think it's in Manual Therapy. And they've been looking at, which percentage of chronic low back pain patients present characteristics that are more, let's say, that fit more into a nociceptive pain, in contrast to neuropathic pain, and in contrast to central sensitization pain, which is currently referred to as nociplastic pain. Don't know if you've heard that term.
Yeah, I've heard that. Yeah.
Which is, it's still not perfectly defined, but it's referred to a pain that may arise from plasticity developed in the nociceptive system. So, it's a bit what we were talking about before. And there's been a couple of large cohort studies and then a few trials also. And they seem to suggest that there's about a third of patients with chronic low back pain that have a pain that can be classified under nociplastic. So I think, if I remember from the top of my head, the different studies go from 25 to 40% of participants in those studies that have, these type of mechanisms likely involved. Yeah.
Yeah, that's fascinating.
And it's also important because we're trying to subgroup these patients, and maybe those patients respond better to specific treatments like spinal manipulation, if this is the way or one of the ways that spinal manipulation acts.
Well, I think that's a fantastic point. One of the huge benefits of these mechanisms is exactly that. How do we classify, could we classify in advance people who are going to respond really well to chiropractic care, to exercise, to nutrition, to, psychological interventions, et cetera, et cetera. That's a great way to actually think about it, I believe. So I'm glad you mentioned that. That's awesome. Mm-hmm. Thank you. Let's get into our last paper here. And this is, when I saw this paper, I knew I had to have you on the podcast. I had already thought about having you on before, but I thought, okay, I just need to call you up or email you and see if you'd come on. So I appreciate you talking about all these things. But this is the paper that ultimately I saw it and I thought, I just have to have you on. So anyways, it's so interesting, so it's called The Presence of Tumor Necrosis Factor-Alpha in Urine Samples of Patients with Chronic Low Back Pain undergoing chiropractic care, preliminary findings from prospective cohort study. So this is published in Frontiers in Integrative Neuroscience. And could you walk us through this one?
Mm-hmm. Yeah, thank you very much. I love this paper too. And, I will say to start with that methodologically is not the soundest paper, but we really wanted to share these results that, as the title says, are preliminary, but, they're, I think, fascinating too, because, we're not the first one to look at TNF-alpha, which is a cytokine. It's a pro-inflammatory cytokine. Other authors have looked at TNF-alpha. There's at least 2 papers by Teodorczyk sorry, Julita, I don't know how to pronounce Teodorczyk Ingeyan.
Yep.
From CMCC.
CMCC, yep.
Which, whose work I really, really admire. And they've been looking at TNF-alpha now for at least 15 years and other cytokines. So what we wanted to do is to see, first of all, going back a little bit, rewinding a tiny bit, patients with chronic pain, and specifically with chronic low back pain, seem to have increased level of a certain inflammatory cytokines, and TNF-alpha is one of the cytokines that pops up, constantly. I don't think it's a biomarker only for chronic low back pain, but it could definitely serve as, a biomarker for this type of chronic pain, among others. So, first of all, we wanted to look at these neuroimmune markers and see if they were elevated in patients with chronic low back pain, and also to see if these markers were responsive to treatment. If after receiving care, there will be any changes in TNF-alpha. The thing is that measuring TNF-alpha in blood is not difficult, but it's definitely more costly. There will be some patients that would not want to do it because they don't want to get blood drawn. You need a phlebotomist or a nurse practitioner to do it. So, we were looking into, how could we do this in an easier way? And we found that in other areas, not in pain research, they had been looking at cytokines in urine, and they correlated very well with cytokines in blood. So we decided to collect blood urines from our patients that were coming to the clinic at the Madrid College here. So this is, I'm talking about this paper as the first author, but most of the work, the idea behind is my co-supervisor, who's the last author of this paper, Arantxa Ortega de Mues. She's not a chiropractor, she's a biologist. She's a specialist in kidney function. So that's why she's like, urine is my thing, I know urine very well. And, we were not sure what we were going to find, but, as this paper shows, we found that TNF-alpha was significantly elevated in patients with chronic low back pain compared to healthy participants. We had a small sample of healthy participants. We're actually working now into increasing the number of healthy participants, but we also know from research in blood cytokines that usually TNF-alpha, if you don't have an inflammatory or a chronic condition, the levels are going to be very close to 0. So, we're pretty sure that chronic low back pain patients have increased urinary levels of TNF-alpha. And we also found that after a period of care, this was not a trial, it was not an experimental study. We, the patients were coming for care with chronic low back pain. We made the diagnosis of chronic non-specific low back pain, and we asked them, do you want to be part of this study? We're just going to measure pain disability, and then we need a couple of urine samples. And that's it. And patients were receiving chiropractic care all of them were receiving SMT in every visit, but some of them were also receiving patient education and exercise. So we're trying to measure, the effects of a pragmatically delivered intervention, which is chiropractic care, not specifically spinal manipulation. Although, the common denominator for every intervention was spinal manipulation. And we found that, TNF-alpha, after an average of 4 weeks of treatment, was decreased in the group receiving care. It was not a large sample. We had 24 participants, but we have a second study replicating these results, where we controlled much better the care they were receiving. They were only receiving spinal manipulation, and actually we're finding even stronger results. It's fascinating. It seems like there is an effect here that may be telling us something.
So curious, has anybody checked TNF-alpha in chronic back pain patients before?
Yeah, definitely.
Okay.
Theodorchik in Jahan, they were looking at.
Oh, they have in the urine.
Not in urine. We were the first ones to measure it in urine. Exactly.
Okay. Gotcha.
Yeah.
All right.
Yeah.
And then, how expensive are these TNF tests? Like, I'm just curious. I mean, I might want to start checking my own patients. I don't know, just to see if it's cheap.
We're doing ELISA, which is, it's not an expensive test. I think all kind of got familiarized with the ELISA test for the antibodies, for the COVID antibodies, for example. So the ELISA is not expensive. We're buying a full kit. It was around 800 euros. So let's say about $900 for, and that kit was enough for a little bit over 20 patients, 22 patients, I believe.
Okay.
So let's say about 40 euros to run one ELISA.
Okay.
These were done in a specific lab in a hospital in Madrid for research purposes, but you could get this done at a commercial level very easily, very cheaply. And this is one of the things that we're interested, like, to see if there's something we could use to predict the response, maybe to evaluate the response as well. But we need more studies, So that's a good point. And I am also, I actually measure my levels before we started. I was one of the pilots, and I was healthy. I had no pain, and I was in zero. So okay, at least it seems to be sensitive to young, healthy participants.
Yeah, no doubt. I wanted to bring back something that you said, that the common denominator in all of these patients was that they got the adjustment or manipulation. And despite, that they had, exercise and all sorts of stuff that was going on. So it makes me wonder, if this, if studies like this could represent a way to identify some of the mechanisms by which chiropractic care would manifest its effects, similar to what we were talking about earlier with the very first study. Is there something specific about this TNF-alpha in the urine that is, what exactly does that tell us? Is that something that this is, I guess the most direct way to interpret it is there's some sort of an inflammatory effect, right? Anti-inflammatory effect. Is that the way you would interpret it? Or
It's TNF-alpha is a pro-inflammatory cytokine that is also, it's the beginning of a cascade of other pro-inflammatory cytokines. So it's one of the reasons why it's important. If you modulate TNF-alpha, other cytokines and chemokines may be modulated as well. And it definitely, it's related to inflammation. So it's pro-inflammatory. We don't know exactly in what stages of inflammation, but there's very interesting work by Klein et al. Who I think they're in Australia, because the lead researcher is Paul Hodges for these studies.
Okay.
They've been looking at also different cytokines in the related to chronic low back pain and the transition from acute to chronic low back pain. And they seem to always conclude that TNF-alpha remains elevated in patients that do not recover from acute episodes. So it could be, what maintains a certain level of maybe what they call now low-grade inflammation. Maybe it's not such low-grade, but it's a term that's used also for a gut inflammation and, for example, IBS.
Yeah.
So, there's something related there to that transition, and also maybe the maintenance of that inflammation and sensitization. It's also important to know that TNF-alpha, this we cannot measure in humans right now, but in the spinal cord may induce central sensitization in animals, or central sensitization-like processes. It is part of the process of what is called neuroinflammation, that you've probably heard of, that has been related to the central sensitization. So there's quite a few ways through which TNF-alpha may induce inflammation in the central nervous system and in the periphery. And we don't know which one manipulation will target specifically. We yet don't know.
Sure. But it's a starting point for sure.
Mm-hmm.
And
for sure.
and what you said about the central sensitization, and that was exactly what I was thinking about was, how are the mechanisms similar or different in those? It would seem, again, I'm going to hypothesize here, but if at the spinal cord level, we have perhaps a reduction of the, ramping up of the volleys of information to the brain to say, hey, we got some pain going on here. And then if we're getting that at a biochemical level with TNF-alpha, sort of, leading to a cascade of other inflammatory soup effects or, that sort of thing, then maybe that is, a potential mechanism by which it's working.
Mm-hmm. There is fascinating work on this exactly. So, for example, there is clearly, it's been shown there is a crosstalk between C fibers and immune cells in the periphery and in the central nervous system. So the activation of C fibers, for example, in the central nervous system activates microglial cells to release cytokines. So, in a way, this is fascinating to me. It's like, okay, so this ongoing pain is also activating like an immune response, and this immune response sensitizes us, probably to protect us. But if that volley, as you were saying, of nociceptive input continues for whatever reasons, it may also be upregulated. That's probably not the right word, but like we could maintain that release of TNF-alpha, which could result in maybe aberrant plasticity. So, where does the adjustment, act here? It's still hard to figure out, but there's a lot suggesting that, these mechanisms are targeted somehow.
Yeah. Yeah. And then couple in, like you said, gut, some of the stuff on gut health, exercise, nutrition, all of these known to have, we'll say somewhat similar mechanisms, anti-inflammatory mechanisms.
Yeah.
And then, but those are all first line types of care, lifestyle issues. So it really is exciting to me how we're getting to a point where, I realize it's a preliminary study, but again, the hypothesis that you can derive from something like this are all over the place. I mean, in a good way, to start looking at these mechanisms in some more detail and be able to, figure out, maybe one day, people come in and they pee in a cup and, hey, you're TNF-alpha. I don't know. I'm just thinking out loud. And, hey, based upon this, we think this, that, or the other type of care, or these two types of care, are going to be the best for you. Yeah, who knows? But but that's sort of the way my brain is working now. Is we take these mechanisms a few steps beyond, hopefully in 5, 10, 15 years, and be able to say to people, with some degree of certainty, hey, this stuff is going to help you out.
Yeah, not just your brain. I think, this is exactly what I explained my patients now. We're still collecting urine. It's like, in the future, maybe, we can just check your urine and see if this is suitable for you, or, or if maybe levels are too high or too low. Maybe you want to have, some other type of care. Yeah.
Well, you've thought about it. You had urine on the brain long before me.
I don't know if that's a good thing, urine on the brain, but
you're right. It's not. When I said it, it sounded weird, but you know what I meant. Okay. So what's up next for you? It sounds like some of the studies are continuing on with the type of research that you've done, especially with this TNF-alpha. What else is there on the horizon for you?
So I'm now finishing a large for me, large RCT with 150 participants. We're we're getting close to finish with chronic low back pain. We're measuring some of the same things that we've been talking about, spinal mechanisms, surrogates or behavioral surrogates of those via measuring pressure pain thresholds. We're also measuring more, let's say, less specific mechanisms with some questionnaires, or maybe not less specific, but more like psychological outcomes. We're measuring TNF-Alpha in urine, and we're using, SMT, the old good adjustment, and we're comparing it to a validated placebo and see if these effects are specific or not. This is always it's a challenging thing to do, and it's scary, because you obviously have your hypotheses. You've practiced long enough to think like, okay, what we're doing is specific enough, but who knows? Honestly, the data doesn't seem to suggest that our effects are specific or that they're very different from the placebo. But that doesn't happen just for manipulation. I think any intervention on pain, and specifically on chronic pain, I don't think many interventions have proved, or any at all, have proved to be better than placebo, So,
right. And what, since you brought that up, I hear this all the time. Well, it's no better than placebo. Hey, placebo is pretty good.
Yeah. It is also something that's, it's controversial, but I agree with you. And I think placebo is just, not just. I think that one of the components of placebo is something that, I'm going to maybe be crucified for this by some purist, very scientifically driven chiropractors. But I think it's also part of the chiropractic philosophy of placebo is the mechanisms of the body healing itself, and if we could tap into those mechanisms, as long as, what we're doing is not just doing that, there's nothing wrong, in my opinion. There's nothing wrong by tapping into those mechanisms while also, putting an input on the nervous system and having patients become more active, getting them some exercise, getting them to understand their pain better. I think the combination is actually great, honestly.
Yeah. And you're right. I mean, there's value in these words, and depending on how you define placebo, but I'm with you. I'm basically, my thought is I'll do whatever it takes, that's ethical and effective to get people better. And if it involves some placebo, I'm down for that.
So, I hear you. Well, as long as it doesn't involve urine in the brain, we're fine.
Okay, well, all right. Yeah, you got me there. Okay, can I retract that now? Now I'm thinking about that.
This is going to stay in the internet,
It will stay forever. Yep. Okay. Yeah. Oh, well. So, Carlos, a goal of this podcast, as you know, is to help to motivate and assist practitioners and students alike to pursue research careers in chiropractic science. And I ask this of all my guests: can you offer any advice to aspiring chiropractors or students who may wish to become these future scientists?
I think this is a great question, and I've been thinking about it ever since I knew you were going to ask me this question. So, but it's hard also. I just want to encourage them to do it. I think there is such a need for research to be done on all the manual therapies. But if we're talking about specifically, a profession, because this is a chiropractic science podcast, get into the science, start reading the cool papers, and try to collect data. If you're already practicing, try to collect data and see how fun it is. The truth is, it's very fun, but it's also super humbling. You have to be ready to accept failure, ready to accept that your ideas will be challenged, and they will be. You have to be ready to accept that you may be wrong, and be flexible enough to change the way you think, which may eventually affect the way you practice. And there's nothing wrong with that. It's the best, I think, the best humbling exercise that any healthcare professional, or actually any professional, could do. I think we should all do that. So get into it from the beginning. Like read the papers, go to these congresses. Not just, seminars are fun. You can learn a lot of technique and work with the gurus. Okay, it's fun. I've done that, but also try to do something that's intellectually challenging, and I think it will be very rewarding. But you definitely need to be aware of the amount of work that it takes. Not just the physical and mental work, but also the work. As I was telling you before, it's super humbling. Haven't you found that too? That you get into science and all you believe, some of them were super strong. You had some pillars that just crumble because the data or the science don't support that.
Yeah, absolutely. And I really like that advice. Let the data go where it goes, and but you have to be curious. Mm-hmm. And I think that's a theme that runs throughout all of our research, and especially yours. And it's so interesting, but there are roadblocks along the way, no doubt, just like there are in practice. So it's a little bit different. The education's a little bit different going through the PhD, certainly, than it is clinically. Yeah. But I think for some people are interested in that, and that's really who I'm trying to get out to. We absolutely need the practitioners who are there in practice all day, every day, taking care of patients. We need those people.
Of course.
But I'm hoping to, we'll say, convert or get some people at least interested in contributing, even if it's from their practice. That's fine too. That would be awesome. Write up some case studies.
We definitely need that type of data too. There's so much out there that we could use. Yeah,
no doubt. I mean, I read all these, as many case studies as I can, and that helps me formulate my next studies. So we need chiropractors in practice to be able to do this and get interested as well. So, Carlos, thanks for such a
let me just add something.
Yeah,
sorry.
Yeah.
I think you nailed it. I realized that I didn't really motivate, but I think it's very related to where I am right now. And I think last year PhD student is like, you only see roadblocks. It seems like it never ends. So maybe that's why I wasn't so inspirational. But I want to say that even now, and it's been so tough. This is the toughest thing I've ever done, for sure. For sure. We opened the chiropractic college from scratch, pretty much. But this is the toughest thing I've ever done. Just do a PhD. That so many of people like you have done before. But I think it's really, really challenging. But it's also super rewarding. I've never done anything that rewarding. So maybe that's a tiny bit of motivation that I could give people.
Well, yeah, I mean, but it has to be real, and your experience is real, and that's why I love having other researchers on. Everybody's got a different take on things. Everybody's got different advice, and so yeah, totally. I understand where you're coming from. Maybe over a beer or something we could talk about
For sure.
some of these things, but yeah, I mean, there were several times I was thinking about throwing in the towel. I think it's only normal at some point during a PhD to have some of these thoughts. Of course, most of us don't do that because we're motivated to get it done, and it's exciting. You spend all this time and effort on projects, but just like you do in practice, some things in practice lead to great success, and other things you spend a lot of time on, they don't. So, it's the same kind of thing, just applied in a research setting as opposed to practice. So cool. Well, Carlos, thanks so much for coming on. This was really, really a great talk. I've enjoyed myself tremendously.
I've enjoyed so much. I can't even start telling you. Thank you so much. This is the first podcast that I participate in as a scientist, if we can put it that way. So it's been so fun. Loved it, Dean. Thank you so much.
Thanks for listening to this episode of Chiropractic Science with Dr. Carlos Gevers. I hope you learned as much as I did in this interview. And I look forward to bringing you more great chiropractic science in the future.